So I've been thinking about this a lot lately, especially in the wake of Tua and yet another concussion. And I know he's going to come back and play again. I know he will. And I want to talk a little bit, if it's okay, about Walter Payton after his NFL career ended. And he died in 1999 of bile duct cancer. So this was right before they started checking for CTE. Here's before they would know to examine a brain.
I wanted to talk a little bit about CTE, chronic traumatic encephalopathy. I actually did some of my graduate research work in it. And the first thing I wanted to say is concussions are not the primary cause of CTE. You don't have to get hit hard enough to get a concussion to begin to do the damage to your neurons that eventually leads to that encephalopathy. It's actually the repeated medium level hits. So when those linemen are just running into each other every time, or when you see a boxer that gets repeatedly jabbed, there's a minimum amount of force that you have to meet. It doesn't look like hitting a soccer ball or the vibration from an electric toothbrush is enough. But the point I'm making is it doesn't actually take that much force to cause it. It's the repeated hits. Every type of dementia that I've read about is caused by proteinopathies, basically proteins becoming misfolded and aggregating together. And depending on what type of dementia it is, there's always one that's common, and then there's always one that's unique. And starting with the one that's common, this is obviously, it's a neuron, and you can see it's very weirdly shaped. Now, what happens with every single cell in the body, internally within all the cells, there are long strands of protein that are pulling the sides of it into the shape it needs to be in. So obviously in a neuron, you're going to have a very complex internal structure of proteins that are pulling all these pieces into the place they need to be. So the first protein that becomes misfolded is these little blue things, represented by these little blue things, and it's called tau. So within the neuron, these long fibrils are made out of tubulin and tau. Tubulin is basically these little beads, the little green ones and the little red ones, and you can think of them almost as little magnetic beads that stick together. And these tau proteins go in between and kind of act like bungee cords that hook onto them. So those tubulin proteins, they form kind of a rigid structure, and then those tau proteins kind of hold them together so that they can bend and twist a little bit and move around without breaking apart. Well, when those tau proteins start to become misfolded and begin to aggregate together, you basically lose the ability for those fibrils to have any kind of flexibility to them. And then when they're put under a lot of stress, they begin to break apart. And obviously this is a big problem because then those neurons lose those internal structures. The second problem is that these are kind of used as highways within the cell. So if there's a particular molecule that needs to be actively transported quickly from one end to another, there are small proteins that almost have little feet, and they will walk along these fibrils. And on the other end, they'll carry whatever that package is, whatever that molecule is, along with them to whatever part of the cell it needs to be in. So when this starts to come apart, the cell loses both its shape and its ability to transport. So that's the protein that's common to many different types of dementia, maybe all of them. The one that's unique to CTE is this one, TDP43. And TDP43 has a function in basically every cell where it kind of acts as a clamp that aids in DNA transcription. Well, with TDP43, it's a protein that can have two configurations. It can have one where all the water-soluble parts of it, the polar amino acids, have their shells on the outside, so this can float around in water. But if it becomes misfolded, then the non-water soluble parts start to stick out. And it's not a big deal if it's just one. Eventually it'll fold back the way it should be. But if you have many of them, they can start to aggregate together. And when they aggregate together, they basically become useless to the cell. Normally what the cell would do is it would send a lysosome over, and the lysosome would basically consume those misfolded proteins, those little aggregates, and basically redigest them into amino acids. And then those amino acids could just be reused to make new proteins. So the first thing that's going on with CTE is you have multiple places where this damage is happening in neurons across the brain, where they're beginning to lose structure and transport, and you're also beginning to have this aggregation of TDP43 proteins that aren't getting cleared out. These are slides from a brain of somebody who died with CTE, and all these little brown dots are actually aggregations of TDP43 protein. And you can see here where they're zooming in on one of them. It's just a dark brown dot. Ideally, anytime there's misfolded stuff or junk that's sitting in the brain, it should get cleared out fairly quickly. In fact, usually that happens within 24 hours. But in the case of CTE, you have enough damage also to the fluid clearance pathways in the brain, as well as to the brain's immune system, that these just don't get cleaned up at all. Now, with Alzheimer's disease, you have a similar thing that happens with beta amyloid. But Alzheimer's is something that tends to set in when you're pretty far into old age, while CTE starts happening to people when they're teenagers. So this is an image from the NIH. You have 4 stages to CTE. At stage 1, the symptoms show up usually as things like depression and anxiety, maybe a little bit of forgetfulness. But if you look at these stage 1 slides, you actually have to zoom in pretty far to be able to see those misfolded proteins. Now you can see with stage 2, those dots are much larger, and you can even see them from here. And here at stage 3 and stage 4, there is significant visible macroscopic damage to the structure of the brain itself. And looking at this one, you can see that very significant thalamic gap. That is a hallmark of CTE. That the thalamus is a part of the brain that kind of routes sensory input to different areas of the brain where it can be needed, where it's used. So that's why when you hear about folks with CTE losing the ability to spell and the ability to read and the ability to recognize other people's faces, it's damage to that thalamic gap, in my opinion, that's probably causing it. Now, as this notes, this sample came from an athlete, an amateur athlete, not even a professional level athlete, who died before the age of 30. So at the top here is a brain with CTE, and at the bottom here is a healthy brain. This is a whole brain image also from the NIH. You'll notice with CTE that you have this much larger kind of degradation on the frontal lobe on the right than you do on the left. I know this looks like the right, but keep in mind the back of the brain is at the top here. And you can also see where the thalamus has been eroded away. The thing I want to point out, though, is that as far as we can tell, the CTE isn't happening because of injuries at the professional level. Most of these professional players are already showing symptoms by the time they get to the NFL. So better concussion protocols and better pads and things like that, they're simply not going to solve it because it's the moderate level hits that are causing the damage. The real solution is for people simply to not engage in activities where you repeatedly take hits like that. play other sports.